As Jay mentioned, I'm Todd Druley. I'm an assistant professor of pediatrics and genetics at Washington University, and I'm very honored and grateful to be here. I'm going to share with you some of the results right now that have come from my A award. I was one of the first recipients in 2009. So this is population-based rare variant detection in pediatric high-risk leukemia. This should be really informal. If I say something that doesn't make any sense, which I often do, just stop me, raise your hand, shout at me. I'm happy to answer questions. I want this to be, really enjoyable and clear for everybody. So I was asked to give you a little bit about my background. So I'm a Midwesterner. I grew up in a little farm town in Illinois, Princeton, Illinois. It's about 2 hours southwest of Chicago, 7,000 happy people in the middle of the corn. I was given a scholarship to go to the University of Illinois for a bachelor's degree in cell biology. I graduated there in 1994, and then I was invited to participate in the MD PhD program at the University of Illinois in Chicago, and that was from 94 to 2002. And in 2002, I moved to Washington University in St. Louis, where I did my residency and my fellowship, and I've stayed on as faculty since 2008. My wife Debbie is an attorney, and we have 2 boys, Eric and Josh, who are 7 and 5. So that's sort of me. And so my inspiration, how did I get into this field? Everything sort of coalesced around the same time. It was in the late 90s during my PhD years doing research. On a personal level, my cousin, who's a few years older than me, had a 3-year-old son who was diagnosed with AML. He was treated at Riley's Children's Hospital in Indianapolis, and I would visit him on a few occasions. And it really struck me how their pediatric oncologist was really an extended member of their family, someone that they knew on a personal level, they relied upon, and really made an impact even after things unfortunately went south. My cousin's son relapsed and passed away, and even after that happened, I saw how much their pediatric oncologist meant to them. And so, I thought, that really struck me. And so at the same time, I was doing more general PhD research in chemotherapy resistance in cancer, and so I was seeing cancer from a more professional side. And then as I got into my clinical rotations, I just really enjoyed being around the children. It keeps things very light and fun, and you feel a good sense of satisfaction working with the children. And all of this came together, and I'm really blessed to have the opportunities that I've had. So now a little bit about the research. So the proposal that I put forth to eliminate STIN sort of is phrased like this, and I'll explain this. So the individual combinations of congenital or acquired rare genetic variants will predispose to pediatric leukemia and either initiate leukemia or change the way the chemotherapy agents act in that particular person. So what we're talking about here is specifically rare genetic variation, and this means genetic changes that are in less than 1% of people worldwide. And why is this important? Because over the last 20, 25, 30 years, researchers have been looking for that magic bullet. What's that one gene or that one genetic change, that one mutation that's really going to explain cancer? And I don't mean to be a skeptic, but we haven't found it, and it's just not there. So this is sort of a clinician's nightmare, because this means that every person has their own personal genetic profile of changes that really dictate how that person at that time in their life under those conditions developed cancer and either responded or failed to respond to treatment. But we can do this, all right? Technology has gotten to the point where we can try to understand what's there. So we know that there are common genetic changes that we see often in cancer. The Philadelphia chromosome, MLL translocations, other translocations. But now that sequencing technologies have advanced, we have plenty of examples of people who have these genetic changes but are perfectly healthy. So that tells us that they are important for influencing the cancer or the survival or the treatment for those children, but they're not the only story. So it really depends in the context into which they've been introduced. If I go start a bonfire in the middle of the desert, it'll burn itself out and not much will come of it. If I start a bonfire in the middle of the forest, the consequences could be catastrophic. So what is that background? What's that environment that's there? And that's something that we want to try to understand. Now, if we're going to be able to do this, we have to survey a huge population of people. You can't pick out a handful of people and say, well, this is unique to this person, and unique to this person, unique to this person, so it applies to everybody. You have to look for very subtle trends across populations of individuals. We need to compare many different genes. There's not one magic bullet gene. This is a subtle interaction between different genes that act and fail to act at different points during development and normal human life activity. And then ideally we have to compare that against an unaffected population of people to find what's unique to the people that have the illness. So this is a cartoon that I use to explain rare genetic variation. So here in scenario 1, there's two main ways that rare genetic variation can influence a particular disease. And so in scenario 1, we have our unaffected population of people. So what we show here, this line is a gene, and these little boxes indicate the parts of the gene that become proteins and have some effect in our cells or our body. So this yellow line is a genetic change that's very rare. It's less than 1% in this unaffected population of people. But if I go and I handpick a group of people, I may find that that same genetic change is now common for whatever condition I've picked for. So an example for this is sickle cell disease. So the mutation that causes sickle cell disease is rare worldwide, but I can go and pick any group of people in sub-Saharan Africa and probably find that mutation at 20-25% frequency. Now, that's not really what we're trying to identify in our lab. What we're trying to do is something a little more subtle and a little more computationally tricky. So this is scenario 2. So we have the same gene, we have the same genetic variant, it's still just as rare. But now if I go and I pick a group of people that have some particular condition, I don't see an increase in that yellow genetic change. It's still just as rare as it was before. But what I've done now is pick a group of people that have a bunch of different genetic changes. Any one of them could influence that gene in a similar way to the yellow one. And so it's this aggregation of genetic change across that entire gene that we could only appreciate if we looked at an entire population of people. So an example of this is cystic fibrosis. So there's a genetic change called delta F508. People thought that was the cause of cystic fibrosis for many, many years. So if somebody showed up with symptoms of cystic fibrosis, they would be tested for delta F508. And of course, right away, people started to have symptoms and didn't have the delta F508 mutation. So now that we can sequence the gene that causes cystic fibrosis, we have a library of over 1,100 genetic changes, almost all of which confer some type of cystic fibrosis. So that's what we're trying to document here in a much faster, more efficient manner. So you can't read this, and that's okay, you don't need to. So what we proposed to do was handpick a group of genes that we thought would really give us a lot of information about pediatric cancer. And we could create a population of children that had high-risk leukemia, and we could sequence all of these genes at the same time in a very fast and efficient method. The reason we picked these genes, and there's 55 of them, and they comprise a broad variety of different cellular and important cancer-related functions. So they were chosen, almost all of them were chosen from previously published literature suggesting that they have some impact on pediatric leukemia. So these are the genes that we chose. So during my postdoctoral work, before I started this, we were working on how do we do these kinds of assays in a more efficient, cost-effective, time-effective manner. And instead of just sequencing one gene in one person, one at a time, one at a time, one at a time, that's a huge amount of time, expense, cost, and effort that it takes to acquire all that data in a sequential order. So if we just take the people's DNA and we just put it into one common pool, and then we survey all those genes at the same time, now we get a catalog of all the genetic changes in that particular pool of DNA samples for all of the genes that we survey. It doesn't tell us which specific person has which specific change, but it does give us an indication of all the genetic changes in that particular group, and we can compare different groups. So we made 3 different pools. We had children with high-risk leukemia, we had their non-cancer DNA. We had the same 96 children, and these children were collected from Children's Oncology Group Protocol 9906, which is a high-risk leukemia studies in the late 90s, early 2000s. And so we had the same 96 children, we had their leukemia DNA. And we compared those to children's DNA that were not affected. And these were taken from Guthrie-Card blood spots. So when babies are born on day 3 or 4 of life, they have their heels stuck and the blood's blotted on filter paper. You can collect DNA from that filter paper and use that for genetic surveys. So there's 55 genes in each of these 3 pools, and we're making comparisons. So we can see what the children are born with, what's new, particular to their cancer, and then what's different from children that don't have these leukemias. So to get to the punchline here, we did all the sequencing and all of these genes, and it's a lot of data. There's 4,000 or 5,000 genetic changes in every pool. And so how do we know which ones are of value to us and which ones aren't? Well, I'm not a statistician, and I believe you can make stats say whatever you want them to say. So instead of doing stats, we created maps of the genes that we sequenced. So what you see here are the white boxes that we sequenced for this particular gene, which is involved in energy and drug metabolism. These are the regions that we sequenced in the white boxes. And we just put a tick mark everywhere we found a genetic change in either our control unaffected population, the non-cancer patient DNA, or the cancer patient DNA. And we said, are there any genes where there's regions of genetic change that's specific to the leukemia patients compared to our controls? And so here, in the red oval, you see that there's no genetic changes whatsoever in this region of this gene in our control population. And yet we have a whole variety of genetic changes in our patient population. All of them, except for this one, the children were born with. It's not something that they acquired through DNA mutation. They were born with these. So the next question is, does that mean anything? Does it have any impact on disease? How do we know if that's related to leukemia or not? Well, we don't, but one proxy we can use is conservation. So that a lot of our genome is the same from human to human as it is to chimpanzees, to rhesus monkeys, and you can compare different mammalian species, and if a piece of your DNA is important, it will be preserved through all of those different animals because it does the same important critical function. So we then laid this track of DNA conservation below here, and so the higher the line is, the more conservation there is at that particular part of the gene. So I put a black rectangle here around this exon 8 in this gene, that's region number 8 in this particular gene. It has a high level of conservation, so this is an important DNA sequence. And yet we see a lot of genetic changes in our patients that we don't see in our controls. So this is a proxy for these genetic changes having some functional impact on how this gene works. And so, we were excited to see that for this particular gene. And so we looked at 2 other energy and drug metabolism genes. And this one is a different gene, and it has the same genetic pattern. There's no genetic changes here in our controls. There's plenty of genetic changes here in our patients. And a lot of these regions are highly conserved. But now this gene here, there's no such pattern. The genetic changes are pretty equivalent between the patients and the control samples. What's interesting about that is that the 2 genes that I showed you that have these patterns were the 2 genes that were significant in the published report where we got these gene lists from. This gene was not significant. That was from a Nature report out of St. Jude's in 2007. So our findings were matching theirs, but we were looking for something totally different than what they were looking for. So do these genes have any potential role? And yes, there's already published literature suggesting that common changes in these genes, not rare changes, but common changes, are associated with an increased risk for developing leukemia in children. Okay? And a lot of these changes are specific to certain ethnic or racial groups. However, we're demonstrating this as in rare genetic variation across an entire population that spans multiple ethnicities. So, the punchline to this story is that it's good to work with the Children's Oncology Group because they have a lot of existing information. It can be disadvantageous to work with the Children's Oncology Group because they have a lot of existing information. So, my good friend Rick Harvey, he's a statistician at the University of New Mexico. He's a wonderful guy. And so I asked him, he studies the protein expression patterns. So not DNA, but the proteins that result from the genes. The genes are telling us how to make these proteins. So he has DNA he has protein expression data from across the entire genomes of all of these exact same children. So I said, Rick, could you give me, this is called a heat map. So if it's in bright red, that means it's a protein that is overactive, and green means it's a protein that's underactive. So I said, Rick, can you give me a heat map that shows all of the patients that I'm looking at, all these children across one axis, and all the genes that I've sequenced on the other axis? And can you just tell me what you see? And I didn't give him any gene names or anything else. I just said, just plot it out and tell me what you see. And he said, Todd, 90% of this is sort of typically what we see. We know what we're looking at. There's common genetic changes in these different places. This is all bread and butter stuff that we've seen for years. However, there's a group of patients over here in the far right column with the question mark. This group of patients died very early in their disease course. They were able to get into remission for their leukemia, but the leukemia came back, and it was particularly aggressive, and it did not respond to treatment. And so they have a different protein expression signature than the rest of these children. They have a massive overexpression of 12 or 14 genes, and they have a loss of function for the remainder of most of the rest of the genes. And he said, I don't know what you're looking for, but here's the dozen genes in this red box. And the top 2 genes in the red box were the same 2 genes that I showed you with those patterns that we're looking for. So this would be the first time that we're able to link rare genetic changes across the population to specific outcomes for children who did very poorly in their treatment course. So the conclusions and implications from this, I think there's multiple, but these are the main ones for right now. One is just being able to show that if you look for aggregations of rare variants, you can find subgroups of children who could be predisposed to poor outcomes. Now, these energy metabolism genes, it's not clear right now if these are causing the children to get leukemia, or if a child gets leukemia and is unlucky enough to have one of these genetic changes in one of these genes, they simply don't metabolize the chemotherapy appropriately. The chemotherapy that we deliver is never active in the way that we deliver it. Your body has to change it into its active forms. And if you can't do that, you're essentially untreated. So that's going to take more investigation. But what can we use this information for right now? So if we can validate this, and I think we will, we'll be able to show that this is legitimate. There's multiple lines of evidence suggesting that that's true. So right now, as a pediatric cancer specialist, if a child came to me with this gene expression pattern or protein expression pattern, I would say, let's skip the 2 to 3 years of highly intense chemotherapy. It's unlikely to be effective for you. And let's go directly to a bone marrow transplant while you're healthier.
Plug on that. Thank you, Doc.
And so we can get you into the transplant while you're still healthy. You haven't suffered all the toxic side effects of chemotherapy that's unlikely to serve you well anyway. That would be my first recommendation. I think there are certain chemotherapeutic agents that are more likely to be dysfunctional given these types of gene changes. So, we're talking about the anthracyclines and the epiprophyllotoxins as 2 classes of drugs that are heavily used for leukemia therapy, which could be rendered inactive by these gene changes. So in lieu of that, you could simply take those drugs out of that child's treatment profile and replace them with drugs that don't interact with these genes and hopefully augment their survival likelihood. That's going to take a lot of prospective clinical trials to figure that out. And then in the lab, we want to try to understand what's actually happening in the cell in the presence or absence of these agents, and see if we can, if there may be existing agents that would be suitable for these gene changes now, or we could potentially develop those. So our to-do list here is to confirm the initial results. We're working on doing that. We're expanding the initial results. So technology's advanced since 2009. So we have another 250 matched patients from the next high-risk leukemia trial, and we're just starting to sequence those. So instead of our 55 genes, in addition to our 55 genes, we're adding another 1,700 genes to look at every gene that we think is involved in energy and drug metabolism. There's a lot of data available for some of these children through a national project called the Target Project. So that allows us to save time and money by looking at data that's already available. Rick Harvey is going to do the multivariate analysis. So we want to know in that little cluster of patients that did poorly, was it a specific gender? Was it a specific ethnicity? Was it a specific age? Was there anything clinically unique about that group of kids that could give us a clue that these are children that should be tested for this pattern early on? And of course, we want to get our data published, which I think will happen before the end of the year. Now, more long-term things to do is to establish model systems where we need to study this in the lab, either through cell culture or animal models. We need to characterize how these genes actually change cellular dynamics. And then hopefully, as I mentioned, we could get this into prospective clinical trials. Now, any questions about that? I'm just going to show you a few slides on an ancillary project that's come out. Yeah, Jane.
Group 1, was that blood block cards from when they were born?
Group 1, the control.
No, the control group was group 3, right?
Yeah. Okay.
Group 1, you said it was their non-cancer cells. Where did you get those from?
So in some cases, they did cheek swab cells at the time of diagnosis because they enrolled on the trial. In some cases, it's a remission DNA after the first month of induction therapy.
Would it be interesting to go back and look at their block cards to see if they had the same?
It would if we could
screen?
get them. It's become sort of a political hot potato going back to these, even if the parents were to give permission to go back and get those, which would take a lot of red tape to get.
Because then you can see if something changed, right?
You absolutely could. I agree with you completely. But the states are the ones that actually own the cards. So the minute the state bureau opens it up to one line of investigation, they're going to open it up to everything, and it becomes.
But did you get them for group 3?
We had them for group 3. One of my colleagues, who's a neonatologist, has, in more favorable political times, was able to get 10,000 of them out of the state of Missouri, and we have full approval for those. So we use those quite a bit. So just a few comments on infant leukemia. So this is a project that has been born out of the work that I showed you on from the Alex's lemonade stand. And so we were approached by Professor Julie Ross at the University of Minnesota. She's an epidemiologist there. And so she's really trying to understand the genetics of infant leukemia. So if you're thinking about high-risk leukemias, infant leukemia is the highest of high risk. Half the infants do not survive. The therapy is so intense at such an early age that the survivors are often left with severe complications in cognition and their growth, their endocrine function, a lot of defects that they have going forward. So she had done a lot of work collecting DNA samples from mothers and their infants that developed leukemia, and was looking at, from an epidemiological standpoint, to say, was there any exposures that these mothers had during pregnancy that could have predisposed their children to developing these cancers? And didn't really find much. There was a little bit of evidence about these topoisomerase II inhibitors, but it wasn't very compelling, and she concluded that a genetic analysis was going to have to happen to make a stronger connection. So approached us and asked if we could do what we call exome sequencing. This is sequencing all 25,000 genes that humans have in 12 pairs of mothers and infants with ALL and 13 with AML. All right, because we didn't really have any data to suggest picking some subset of genes, so we just went after all of them. So what we're trying to do here is you have the infant exome. So you've got genetic information on all 25,000 infant genes. And by sequencing all the genes in mom, you can see which genetic changes mom passed on to baby. Flush. And so then you can compare that to the whatever's left behind. It's really paternal, but I can't exclude because I don't have any dad's DNA. Yeah. So I can't say for sure that what's left behind isn't new genetic changes just in that baby. So I've called it the non-maternal exome here. And so you look and see, if I shuffle these cards, how do I end up with the deck stacked against the baby? And that's what we're trying to do here, look for these combinations. So this is early, this is still very preliminary, but if you say which mechanisms, which genetic pathways are most frequently disrupted when we do this, there's a few here that we see come up time and time again. ABC transporters, this is a type of protein that's very important for movement of different electrolytes through the cell, a variety of developmental defects, and some other mechanisms here that are more involved in gene regulation that we're seeing come up. We're about 75% through the sequence. Now, what's also interesting is there's a type of DNA, or excuse me, RNA called microRNA, and so these are little tiny pieces of nucleic acids, and they turn on and turn off huge subsets of genes and really regulate a lot of different genes. And we're seeing that certain ones of these are unique to the changes in the babies with ALL or AML. A lot of this has to be validated. It's still very preliminary, but this is brand new evidence for a role for these. All of these have been implicated in adult cancers, but never in pediatric cancers. So we just have to finish the exome sequencing here, sort of refresh our bioinformatic analysis and see what we're left with. And then longer term, we actually have access to triads where you have mother, father, and baby, and we can start making more precise genetic pathways. It really looks like right now, one parent has a genetic profile that looks as if it's, if you just sort of put it into an agnostic bioinformatic system and say, all of these genetic changes are most likely associated with what? And a lot of times it's just sort of general cancer. There's no specific type. Whereas the other parent looks like the genetic changes that get passed from that parent to the baby are more specific for leukemia. Okay? And so it takes the combination of the two, because it's absolutely unheard of for the parents of these infants to ever get leukemias. And so it just doesn't happen. So you have to just shuffle the cards in the right way, and then we'll see if there's any new genetic changes that get overlaid on top of that profile.
So you don't see where kids that get leukemia, the parents don't get leukemia?
Almost not. It's unheard of. I mean, you can have, even in families where you have the cancer-causing genes, you think of like BRCA1 genes in breast cancer. The definition of an early onset cancer is before the age of 50. And so Christina Applegate, right, a few years ago, elected for a double mastectomy. She was BRCA1 positive. She had breast cancer on one breast. She elected for the double mastectomy, knowing her genetic risk. That happened when she was 26, 27, 28 years old. Mm-hmm. So, other families where they have genetic changes in P53 or APC, they still don't get cancers until they're late teens at the earliest. Most often it's in their 20s. So the reason an infant or a 4-year-old gets a cancer is it's completely separate from that, and we don't really understand. My lab, thank you, of course, to Alex's lemonade stand, which did the heavy lifting on this project. A little bit of help from the National Cancer Institute. And Seth Matthews, Leukemia Foundation. So thank you for your attention. I'll take any questions.